Okay, so the next presenter is Dr. Daniel Etongo. His research interests include socio-ecological system modeling, biodiversity assessment, vulnerability assessment, climate change, adaptation, and mitigation. He represents UNICEF on the Seychelles National Climate Change Committee and contributed towards the development of Seychelles climate change policy. Dr. Etongo, we are good to go.
Okay, thank you very much, Francesca, and I'll just share my screen. So right away, I'm going to be presenting a research work that we did on growth rate and survivorship of 3 mangrove species in the Seychelles under different treatment conditions and how this can have implication for habitat suitability and some kind of restoration initiatives. Just a little bit of information. This paper is currently under review with the Journal of Coastal Conservation. So please keep an eye on this if you want to have a copy of the final product. And to have some little acknowledgement, this work was done through the EBA South, what we call the EBA South project with the then Ministry of Environment and Climate Change. We know there has been this change due to new restructuring. And the PS for Energy and Climate Change, PS Wills Agricole, was the direct executing partner with the wetland unit. So my presentation, the outline, I'll have a very brief introduction, methodology that was used in the study, some of the key results, discussion, and some concluding remarks. So for Seychelles, I will not go much into the aspect of what are mangroves, but for a country like Seychelles, going by the Seychelles Blue Economy Strategic Policy Framework, mangroves are very important, not just as a buffer, but also because Seychelles depends a lot on its blue economy for economic development. And the need for mangrove rehabilitation gained this kind of momentum in 2006, when IUCN and the United Nations Development Program had to develop the mangrove for the future initiative. And they always do this because there was a tsunami around that time. And mangrove are also important for disaster risk reduction. And of course, mass production of mangrove seedling and successful germination of propagules are very essential component in terms of rehabilitation. Because why? The mangrove itself provides us with essential goods and services in terms of ecosystems. However, we have to get this kind of greater understanding on how the propagation should be done in any kind of rehabilitation program or initiative. And of course, it is not a kind of like black and white that mangrove, they can really self-repair themselves. So some form of human intervention is really important to ensure that the restoration initiative is successful. But this is not a kind of straightforward process because there are some challenges. And the challenges, they can range to some kind of technical issues, to issues that emanate from the ecology of mangrove. We have to understand how the ecology looks like, especially from one species to another. There is also this challenge of lack of long-term monitoring program, because whenever we engage in this kind of project or initiative, there is this kind of planning phase, implementation, and also monitoring. And of course, there are some data gap and the lack of baseline studies from which we can draw some best practices. So the objective or the aim of this study was to assess the growth rate and survivorship of 3 mangrove species. So Rhizophora mucronata, Avicennia marina, and also Cereops tagal. And they were irrigated under different conditions using fresh and seawater. So there were 2 sites for this particular project. The Answire mangrove was one of the sites. And in this site, we have approximately 3 hectares of mangrove coverage with 3 species. But then we have also the Port Launay site that was also selected. So you can see that the site A is Port Launay, and then the site B is the Answire mangrove. And for the Answire, the focus was more on the Avicennia marina, while Rhizophora micronata was not that much at the Answell site, and then the Cereoptagal was not even evident there. So they need to collect just one species at the Answell site. And for the experimental design, so 5 liters bottle were cut at some 19 centimeter height, and they were used to pot the seedlings. And in total, there were 65 liter bottles. So 60 in the sense that each of the species had 20 liters, 10 for the fresh water, and then 10 for the seawater. And of course, there was some kind of species identification. So they were being labeled so that we can easily identify which of the species and under what condition they have been treated. So this is a photo of what was done in the experimental design of the work. Then for the collection of the propagule, I will start with B, which is a Rhizophora mucronata. It's important to understand the fruiting season of these propagules. So for Rhizophora, it gets this kind of yellowish cotyledon when it's very close to its fruiting season. And then for the C, which is also the Cereoptagal, is same. It also gets the yellowish cotyledon. While for A, which is Avicennia marina, you have also what we call the pericarp or the seed coat. So it gets also yellowish, and it's much more better to have it when the seed coat is even breaking out, because this is a time that it performs better when being planted. So these were the information gathered in this experiment. The root, they were being measured, the leaf, and also the stems, they were being measured with a ruler. That is an example of what was done with the Avicennia. And if you look at the figure C, you will see that the cotyledon is somewhere in between, and the stem goes from above and below. So it means you have to do the measurement above and also below, because at some point, the cotyledon will fall off, because you have to consider that the stem starts from where the root emerges. Then some of the results in terms of root initiation, how long did it take for us to see like the very first root in these three different mangrove species under different conditions? You see that for marina, for Avicennia marina, it took an average of around 7 days for the root initiation. But for Rhizophora mucronata, it took the longest day. So when you look at what we did with the ANOVA test, for Cereoptagal and also Avicennia marina, there was no significant difference because it's like greater than 0.05. But then for Rhizophora mucronata and also Avicennia marina, there was a significant difference. And so also was the situation with Cereoptagal. So when you look at what happened with the root initiation, it took far much more longer time for Rhizophora to have their first root initiated. So what about the leaf? It's same also for the leaf, because for Avicennia marina, it took much less time compared to the other two species. And the same thing also with the statistical test, which was significantly different for all three species when you try to compare them. So for the root count, this was done within 6 weeks period, because why? After 6 weeks, they began to get entangled. And each time you take the roots for measurement after 1 week, you have to replant it also, which might affect or influence the result. So after 6 weeks, we had to stop at that point, because why? They were entangled that they began to break. So you will see that the result for the leaf goes up to 12 weeks. I'm trying to explain that so that you don't get confused when you see 12 weeks for the leaf, because they were above the ground and they were not affected the way the roots were affected. But having a look at the root, I will just point some few issues that you will see. When you look at the B, which is the Cereoptagal, the root did not show up even like until the 2nd week. And the same thing also with the Rhizophora mucronata. But with Avicennia marina, it started even from the very first week. So you could see that there were differences in terms of the root growth in terms of length. Then for the leaf count, for Avicennia marina, the third week already, you could already see that the leaves could be counted. Why for Ceroptegal, from the fourth week it started, but then for Rhizophora mucronata, it was until the sixth week that you could see. But having a closer look between what happened with the fresh and the seawater, for Avicennia, it was much more better in fresh water than in seawater. Then for also the length of the leaf, you could see similar pattern repeating itself. For Avicennia marina, it was until the third week already that you could see the length of the leaf, and it was also doing much better in the fresh than in the seawater. But when you look at the sea graph, which is that of the Ceroptegal, so the tegal shows a pattern that from the fourth week, it is consistent. And it was much more better also in the fresh water than in the seawater. What are some of the key issues when we have to begin to interpret this result? So the first thing that we had to look into was the effect of fresh and seawater during propagation for mangrove species. So the main root initiation days between Rhizophora mucronata and Avicennia marina, and between Rhizophora mucronata and sea tegal, did not differ when they were compared. But why the main root initiation days did differ when we had that of Avicennia marina and also with the sea tegal. Why in terms of root count, our mucronata recorded the highest number in both fresh and seawater treatment, while A marina dominated regarding root length in both treatment groups. So we also look at the growth response of mangrove species in fresh and seawater treatments. So there were apparent differences in A marina species in comparison to the other two species when they were in different treatment conditions, so the fresh and the seawater. So Avicennia marina is a mangrove species that grows much better in fresh water when under nasal conditions. So this can be due to A marina being a pioneer species with the ability to cope with and adapt to changes in its environment. So it's not really affected by this kind of salinity gradient. So there are some studies that have been supporting this fact, like that of Lee et al. In 2016, that the Avicennia marina is something that is not affected by salinity and also aridity gradient. And also it goes further to explain that even under unfavorable conditions, such a fluctuation in salinity, Avicennia marina is still able to exploit the situation and it grows faster than other species. So when we look at the situation, our result shows that freshwater as opposed to seawater is more suitable for irrigation of mangrove seedling. And we can take also advantage from rainwater harvesting. So we can harness that source of water and it becomes a kind of win-win situation. And these findings also, they can be beneficial when we are designing this kind of nursery for mangrove restoration project, which is something that it saves time and labor when we compare what seawater irrigation can bring into the picture compared to fresh water. So based on our finding, mangrove seedlings do have a slower growth rate in seawater than in fresh water. And this is sometimes species specific. It is not black and white. And it was highlighted by the A marina species. So the replanting, each time we did the measurement, could have also had an influence over the results. So some concluding remarks. So this approach can also help to be able to know that why we engage in restoration, we can decide which species can do much better. And of course, the aspect of zonation cannot be ignored, that mangrove species, they do much better in terms of different salinity gradient. One important point that I want to also highlight here concerning the result, we couldn't do this in the open because rain or some kind of tap water to irrigate as fresh water can have an impact also, because why? The tap water has chlorine in it. And of course, the A marina is not constrained by fresh water as it did grow faster. An important point that I got from the Kun Pisit, who is the co-founder of the Thailand Yatphone Mangrove Preservation Project, he said, mangroves are like the kindergarten, while seagrass are the secondary schools and coral reefs are the high schools and colleges for fishes. And once the fishes graduate from university, so such a statement does highlight the importance of mangrove, especially in the context of Seychelles, in which over 70% of the original mangrove coverage has been lost to land reclamation, road construction, or infrastructural development in terms of the critical infrastructure development in the Seychelles. So these were some of the references I used in the presentation, and thank you for listening, and I welcome your questions.
Thank you, Dr. Etongo. I was going to ask you a few questions, but I think your conclusion sort of gave me the answer, because I was going to ask you, what was going to happen now that your research is over? What is the next step? That sort of thing.
Yeah. Okay, this was part of the EBA South project, and what the University of Seychelles is still doing is ongoing monitoring of the mangrove at Answaya. And it is a kind of learning by doing process. And most of the time, Terence also tried to collaborate in this restoration initiative. Last year, we had some mangrove planting, which was quite good.
Okay. You also mentioned that there's some stakeholders that will benefit from this research. Are you at position now to tell us who these stakeholders are, or is it still something that you're in discussion?
Yeah, there are different stakeholders. What we are going to be doing maybe at the beginning of next year, we want to design a community best practice for mangrove restoration handbook. And we can use this also to share with the responsible stakeholder. The blue economy is one of them. We have the wetland unit that we were working with in this project. And also we have also some of the research NGOs. They should have access to the Life Club of Seychelles also, so that we can reach to the younger generation. And this, this handbook is being financed by COMESA, the Common Market Area for Eastern and Southern Africa. So that is one way to ensure that the knowledge system is sustainable.
Yes, it's continuous, yeah. We do have 2 questions from the Q&A. One of them, do you know how to do these species? Do you know how these species do in the salinity levels between fresh and seawater?
Okay, I can see the question. Do you know how
you see them? Yeah, okay.
Yeah. So what I presented as the result were the 2 different conditions of the 3 different species. And when you see the result, Avicennia marina does much better even in the seawater condition as compared to the freshwater condition. But this was not the case with Rhizophora mucronata and also the Ceroptega. So it means that fresh water and also seawater do have an influence, especially on these other 2 species, which is the Ceroptega and Rhizophora mucronata.
Okay, so the next question, I think you can see it, but I'll just read it out. So going forward, would nurseries use fresh water only for seedling, and how resilient are the seedling once transplanted?
Okay, when I mentioned the aspect of the ecology, we also have to think of the health of the seedling. Do we have some seedlings that have been affected, and then when you try to plant it, the success rate is affected? So we have to also consider that. So that is one issue. But then fresh water has shown in this experiment that it's much better. And I did mention the aspect that it is both labor and also time efficient. And of course, we have a lot of rainwater, and trying to use seawater to irrigate the nursery might be quite challenging. So we just need a rainwater harvesting system, and we can improve on our nursery condition, which is much more realistic.
Yeah, very interesting project there, Dr. Etongo. We'll move on to the next presenter. We'll just keep you here for the end if there are any extra questions. Thank you very much.